All right. Well, today is the day of peptides, proteins, and amino acids, which is kind of a fun subject and has been sort of my area of research for a long time, and so I tend to geek out on it. We're going to just start with the beginning, and some of the things that we talk about will be in the text, others will not. I'll try to make it clear what's important and what's not. In any case, you're going to hear the term peptide, and you're going to hear the term protein, and all of these are polymers of amino acids. And there's really not a great distinction between length in terms of what's a peptide and what's a protein, so I don't even try anymore. Generally, peptides are thought of as less than 50 amino acids as a rule of thumb. Proteins thought of as maybe greater than 30 amino acids, so there's some overlaps. Overachievers call them polypeptides. I laugh at that because peptide itself implies polymers of amino acids, but you'll occasionally hear people talk about polypeptide, and you can laugh at them if you want, although it's unkind. An amino acid is the basic subunit of a peptide or a protein. It is exactly as it sounds. It's got an amino group and a carboxylic acid group and an alpha carbon in between them. People will call peptides composing, well, sometimes people will call these alpha amino acids. The amino acids differ in what's going on with the alpha carbon. All of them have this stereochemistry, and then there's some R group. And the identity of the R group is there are 20 different R groups that nature uses in proteins that are encoded for in DNA and RNA. So you've got sort of 20 different options here. Of course, the organic chemist says that I don't want to be limited by what nature comes up with, and so there are a lot of synthetic amino acids as well that differ in what's here at the R position. There's a lot of different ways you can incorporate those into existing proteins. Another interesting fact along the way is that you can put additional carbons between the amino group and the carboxylic acid group. So, for example, there's a whole class of amino acids that are called beta amino acids. My PhD advisor worked a lot on this. Let's see. Here's an alpha carbon, here's a beta carbon, and then, depending on the identity of the side chain, you could have all sorts of different beta amino acids. Nature generally does not use these, though chemists have started to use these for interesting purposes. The amino acid has, as I said, an amino group and a carboxylate group. Let's see, the text, I believe, points out that if you had an amino acid at physiological pH, the positively charged form of that amino acid side chain, or I'm sorry, the amino group, has a pKa of around 9. That is lower than expected for an ammonium ion. The reason it's low is because of the electron withdrawing ability of the adjacent carboxylic acid. Similarly, the acidic proton of the carboxylic acid has a pKa of around 2, and that's a little bit lower than expected for carboxylic acid, again because of the electron withdrawing ability of the nearby nitrogen. And so your text points out that at physiological pH you'd expect for an amino acid it to be in this what's called Zwitter ionic form. A Zwitter ion is a neutral molecule that has a plus charge and a minus charge in the same molecule, which is okay and interesting and true. Nevertheless, if you look at what the structure of a peptide or a protein is, most of the amino acids in a peptide or a protein do not have charged amino and carboxylate groups. And the reason for this is that the individual amino acid subunits in a peptide or a protein are involved in amide functional groups. And this may clear up something that was a challenge for some folks on the exam 1 question about pKa. So I'm just drawing a peptide or a protein of arbitrary length. And we'll fill in the various side chains. These R groups can be different from each other, so we're just being as general as possible. Notice that we can highlight individual amino acid subunits. Here's one. Here's the next one, and so on. Running out of colors, choose green, pink. Okay. So highlighted are the individual amino acid units, and notice that each has a nitrogen, an alpha carbon, and a carbonyl carbon. And then you go to the next amino acid, nitrogen, alpha carbon, carbonyl carbon, and to the next one, nitrogen, alpha carbon, carbonyl carbon, and so on. The pattern of nitrogen, alpha carbon, and carbonyl carbon is called the peptide backbone. And notice that the amide or the nitrogen of the blue amino acid is, we didn't want that. Nitrogen of the blue amino acid is involved in an amide bond with the carbonyl carbon of the orange amino acid. So that's an amide bond, or in other words, a peptide bond. Those words are used synonymously. Okay, question so far? Fairly boring. Amide bonds, as you've learned, are really stable. They're more stable than ester bonds, so this is one of the reasons why nature uses amide bonds, because they last a long time and they're difficult to hydrolyze. We've talked about making amide bonds before. We've talked about the ribosome before and how these individual bonds are made. So let's see. In general, as I think I tried to point out in the review video for the exam, amide nitrogens are different than amino nitrogens because the lone pairs in these amide nitrogens are tied up in resonance with the carbonyl carbon. So the lone pair on an amide is not very basic, meaning we don't ever worry about what the pKa of the following positively charged amide is, because the nitrogens sp2 hybridize so that the lone pair can be conjugated with the carbonyl group. So in general, when you look at a peptide and you worry about, well, where are the charges, what groups are positively charged versus negatively charged, we don't worry about the amide bonds in the backbone, okay? So again, the backbone is the pattern of nitrogen, alpha carbon, carbonyl carbon on repeat. And then the side chain is what all these R groups are. A couple other features of a peptide. Notice that it has directionality, that is, the two ends are not identical. On one end, you have an ammonium group, or an amino group. We call that the N-terminus. Except there's not space. And then over here, we've got the carboxylate group. We call that the C terminus. And so those two things are different. If you draw out the amino acids, you can draw it in any direction you want, provided the connectivity is correct. When we learn one-letter codes for the amino acids, you must always write the one-letter code for the amino acid in from left to right in the direction from N to C. We'll try to demonstrate at some point how you'll come up with an entirely different molecule if you write the sequence in the wrong direction, okay? All right, any questions so far about peptide structure? We'll talk about what those individual side chains are in just a little bit, but I want to zoom in on one of these amino acids and how best to do this. And I want to talk about conformations of peptides and proteins. So let's take this peptide that we've drawn and sort of blow it up, zooming in a little bit more. Let's start with the bond between the carbonyl carbon of the orange amino acid and the carbonyl and the nitrogen of the blue amino acid. I guess as part of this discussion, sometimes in order to talk about long-range interactions among amino acids, it's helpful to describe their relative spacing. So sometimes we will call the first amino acid in some sequence the I position amino acid. Then we'll call this I plus 1, I plus 2, and so on. That's just, I don't totally know where that came from, but that's, you will probably hear me say things like that, and you may be asking, how do I know which one is I? You don't. You get to say which one is I, and then everything else is relative to that. I plus 1, I plus 2, or I minus 1, I minus 2, or so on. Okay? The other thing that you will hear is people will talk about amino acid residues. The concept of a residue, I'm not totally sure where it comes from or why they chose that word. A residue is just an amino acid within a peptide or a protein. And you can see that, for example, if we zoom into the purple amino acid residue, it's not exactly an amino acid anymore. It's what's left after you make the amide bonds, right? So everything between here and here is the purple residue. Again, you may have to get used to that way of talking about peptides or proteins. So let's talk about this bond between the carbonyl carbon of the I position residue and the amide nitrogen of the I plus 1 residue. And let's just imagine, we could even draw a Newman projection if you want, but let's consider rotation around that carbon-carbon bond. What do you know about amides in general? What's the hybridization of the carbonyl carbon? Sp2, good. How about the nitrogen? Also Sp2. Because of resonance, these atoms, the carbonyl oxygen, carbonyl carbon, amide nitrogen, and amide proton, are all in the same plane, okay? So both of these, let's see, both of these atoms are planar sp2 hybridized. And if we were to draw a Newman projection where we looked down the bond between the carbonyl carbon and the amide nitrogen, what we would see would be something like this. So here's the carbonyl carbon in front, coming from our perspective in the plane of the page, looking down that bond. This is our orange amino acid. I'm just going to write alpha here, and then this would be the carbonyl carbon of the orange amino acid. In the back, we would see, where we can't see it is the amide nitrogen, but coming down towards the bottom of the page, eclipsed with the alpha carbon is the amide proton, and then up here, eclipsed with the carbonyl oxygen is the alpha carbon of the blue amino acid. Okay? You see that? So, and again, this allows lone pairs on the nitrogen to be lined up with the pi orbitals on the carbonyl group so that you can have resonance. The angle between the alpha carbons that's called a dihedral angle, and it's given the Greek letter omega, and here omega equals 180 degrees, or minus 180 degrees. They're actually exactly the same. Because the amide bond has to be planar, there are two possible values for that omega dihedral angle. One is 180, and we call that a trans amide conformation. The other option, not shown here, not drawn above, is a cis amide, and in the cis amide, the two alpha carbons are going to be on the same side. In other words, in the trans amide, you're going to see this, looking at it from the side. In the cis amide, that omega dihedral angle is 0. Okay, and those alphas are just abbreviations for the alpha carbon. Which of those two situations do you think is more sterically demanding? Yeah, the cis amide should be a bigger problem. More sterically hindered. Oh, we can't really. And so cis amide bonds are less common in peptides and proteins than our trans amide bonds. They do occur, and they occur more often for some amino acids than others, and we'll talk about them. All right, so you've got sort of three bonds in the backbone of a peptide or a protein, right? You've got the bond between amide nitrogen and alpha carbon. You've got the bond between alpha carbon and carbonyl carbon, and then you've got the bond between carbonyl carbon and amide nitrogen. So this bond between carbonyl carbon and amide nitrogen is planar and has to be. Let's talk about the other two bonds which do not have dihedral angles that are constrained, like this omega dihedral angle. So let's now talk about the bond between the amide nitrogen and the alpha carbon. And again, we're going to sort of put ourselves in the plane of the page. We're going to look down the bond between amide nitrogen and blue amide nitrogen and blue alpha carbon. And let's see what we would see. So we're looking straight at that amide nitrogen. Now remember that amide nitrogen is sp2 hybridized. So if we put ourselves here looking down that bond, we're going to see towards the bottom of the page the proton, and towards the top of the page we're going to see the carbonyl carbon, which I'll just write with a C. That's okay. Now in the back is the alpha carbon, which we can't currently see, but that alpha carbon has three things attached to it. That alpha carbon is sp3 hybridized, right? So the way we've drawn it now, looking down that bond, it looks like the bond between alpha carbon and blue carbonyl carbon should be eclipsed with the bond between nitrogen and proton. Doesn't have to be that way, but the way we're currently drawing it looks like it's that way. Then we would have an R group here and a hydrogen there. Okay, so in yellow is the carbonyl carbon from the I position amino acid, and then in blue is the amide nitrogen for the I+1 amino acid. Oops. The angle between carbonyl carbons here and here is a dihedral angle as well, and it is given the Greek letter phi. Okay. And there are a lot of different values that phi can adopt. You sort of from 180 to minus 180. Though some values are favored versus others, and that is because if you actually built a model of a peptide or protein chain and started twisting freely around that bond, the phi, the bond between amide nitrogen and alpha carbon, for some values of phi, the carbonyl carbon here starts bumping into things over here. There start to be steric problems. I'll show you an animation of that in just a little bit. So we'll say only some values of phi are allowed. All right, so phi is the bond between amide nitrogen and alpha carbon. Questions so far? Will the R group always be on the right of the Newman projection? Well, it should be wherever, I mean, it depends on, the stereochemistry is always the same. But because you can freely rotate around the bond, no, it doesn't always have to be on the right. Yeah. All right, let's look at now the bond between alpha carbon and carbonyl carbon. That's our other rotatable bond. This is going to be given the Greek letter psi, which looks like a weapon of the same name. Any Ninja Turtle fans out there will recognize the, anyway, sorry. Here's the alpha carbon in front. The alpha carbon's attached to three things. If we put ourselves here and look down that bond between alpha carbon and carbonyl carbon, what are we going to see? Well, here is the nitrogen coming out to our right should be the side chain and then the alpha proton. In the back, you have the amide nitrogen and the carbonyl oxygen. And at least as we've drawn it here, the carbonyl oxygen, let's see, I got that wrong, didn't I? No? That works. at least as we've drawn it here, looking down the bond between alpha carbon and carbonyl carbon, the CN bond in the front should be eclipsed with the carbon-oxygen bond in the back. So just highlighting in blue everything that's part of this I+1 amino acid. And then in purple is the part that's part of the I+2 amino acid. The angle between amide nitrogens here is the dihedral angle that's given the Greek letter psi, except you can't spell it. I can't anyway, PSI. Again, you can have values spanning from 180 to minus 180, but again, only some values are allowed because of sterics. Okay, so the overall shape of a peptide or a protein is determined by these three dihedral angles, omega, phi, and psi. And you've probably learned in other classes about peptide or protein secondary structure. Have you heard about an alpha helix before? I'll show you what one of those is. Those secondary structures are defined by certain values of phi and psi for each of the amino acids. All right, let me see if I can find soap bubbles. Let's see if I can find the a website where you can alter phi and psi and see the consequences. No, that's not it. Nope. Okay. Oh, did I have you? Okay. Thank you. I didn't realize that was in the study guide. Let's see. Bioinformatics slash what? Okay, I'm just going to sorry about that. That'll work. Oh, there it is. Right, so if you've played around with this, I guess it's in the study guide, you can see, oh, it's going to be really slow, isn't it? That's painful. So we're looking down the bond in green between a nitrogen and an alpha carbon, so that's the phi dihedral angle. And you can have it show clashes between potential groups. So for example, we're looking at the amino acid alanine. In the middle here, in gray is the, I guess I need no laser pointer. Use the mouse. Does that work? In gray, can you see the mouse? Everybody can see the mouse, yeah. Is the alpha carbon. Here is the side chain of alanine. And you can see that the side chain is clashing with the carbonyl group of the previous amino acid. So that's an example of a steric clash that makes this particular combination of dihedral angles not so favorable. Now we can press the button and watch that go away. And so now you'd be in an area for, what that means basically is that this conformation where you see steric clashes, those conformations are higher in energy than conformations where there aren't any steric clashes. And you can see that at other values of phi, the carbonyl groups from the two different amino acids start clashing with each other, okay? So that's why only, as I said, only some values of phi are allowed. Now let's find one that's tolerated okay, minus 55. There is a sign convention for how you know whether your dihedral angle is positive or negative. Don't worry though, you don't need to know it because it's all just vector math and it comes out of basically the dot product, I think. Anyway, who knows? But maths will tell you what the sign is. Minus 55 tends to be a good combination because it keeps the carbonyl oxygen away from the two things it can bump into, right? It can bump into the, it can bump into the carbonyl carbon, the carbonyl oxygen from the next amino acid, or the side chain, okay? Get past that and you've got a wide range of values without a huge amount of steric clash because the only other thing you've got to worry about is that alpha proton. So it's sort of like you have this whole hemisphere of rotational values that you can adopt and then that are open. Minus 55 tends to be a decent one. Now let's look for psi, which is the next amino acid, and let's just sort of start rotating. You can see that at some values of psi, we start to have problems not only with the carbonyl oxygen of the next amino acid, but also with the amide proton. So, and then you can rotate around and there's quite a few values that are tolerated. There we've got major problems. But this area, minus 35, minus 55 for both, minus 45 or so for both of them, tend to be okay. The two dihedral angle values that I've shown you here, phi and psi, are actually consistent now with being in an alpha helix. But you can play around with this for a long period of time and sort of see what values are possible. Plotted here is something called the Ramachandran plot, and on the x-axis you have phi values, and on the y-axis you have psi values spanning from plus 80 to minus 80. And each of the dots in the plot represents an observation from a database of proteins. And you can see in the Ramachandran plot sort of what we saw above in playing around with dihedral angle values, which is some of them result in steric clashes, and so they aren't very popular. There aren't a lot of examples for within proteins of amino acids that have dihedral angles of 0 and 0, right? If you look at this plot, there's no examples here. There's tons of examples in this area. There's tons of examples of observations up here, and there's only some here. But there's a lot of portions of this plot where there aren't very many examples at all. Let's see if we can find, let's go back up to this plot, and let's just try to dial in some very unpopular phi and psi values and see if we can tell why they're unpopular. So I think minus 175 is about as close as we're going to get to 0. No? Let's try that again. Okay, 5 might be as close as we're going to get to 0. We'll go to psi. Wrong way. All right. Can you see why there's no examples of amino acids near 0, 0? Because if you do that, look at that. They're like right on top of each other. You can't actually make that happen. You can see space, if we wanted to see space filling, you could definitely see how they can't adopt that kind of conformation. Okay. In contrast, the one that I showed you earlier with like minus 55 puts you somewhere in this region of the Ramachandran plot, and there aren't any steric clashes. So the places where you see the most dots in this plot are areas where there aren't that many steric clashes. And this determines, in a lot of ways, the kinds of structures that different peptides and proteins can adopt. If you have an extended chain of amino acids that all have dihedral angles in this area of the Ramachandran plot, like minus 50 or something phi and psi values of like minus 30 or minus 40, that's an alpha helix. That is what an alpha helix is. If, in contrast, you have an extended chain of amino acids that have dihedral angles of like minus 150 phi and then plus 150 psi, that's going to give you a beta strand, and that's what defines a beta strand. Okay. There's tons more that we could say about that, but that may be enough for now. Any questions you want to ask about that? No, please make it stop. Go ahead.
Where does the tertiary structure reside in that?
Where does tertiary structure reside? Tertiary structure involves association of two different elements of secondary structure. So secondary structure is defined by the backbone angles, the phi and the psi. You're not going to see tertiary structure on this plot. However, tertiary structure can influence phi and psi angles. There are some situations where an alpha helix will actually tighten up a little bit to accommodate packing against another alpha helix, and that can create small changes in dihedral angles. But yeah, this kind of plot basically only shows you secondary structure. What else? I mean, while we're on the subject, you can see this portion of the plot is called the alpha, need a laser pointer. This portion of the plot is called the alpha helix. We will talk about the alpha helix, and we'll talk about why it's called right-handed. This is the right-handed portion of the Ramachandran plot. Over here are dihedral angles that are called left-handed alpha helical. Maybe copy. Let's see if this will work. Probably not. Ah, that's great. So at some point here, you've got a dihedral angle of minus 57 and minus 47, which is for a right-handed alpha helix. And then over here, you have plus 57, plus 47. Those two conformations are sort of enantiomers of each other. And you can see that, what's the best way to illustrate that? Let me dial in the minus 57. Let's get rid of this space. Let's show clashes. So we've got something in roughly an alpha helical conformation here. So if we go now to the, I'm trying to decide whether or not this is worth it, and I guess I basically said yes. Anyway, I guess what I mean is, what I'm trying to illustrate is why there are so many fewer examples of amino acids over here than there are here. And that's controlled by the stereochemistry of alpha amino acids. The amino acids that make our proteins have this stereochemistry where the R group is going into the page. For most amino acids, that stereocenter is going to be S, though that's going to depend on the identity of the side chain. There's another way of talking about amino acid chirality, and that is that these are L amino acids rather than D amino acids. But in any case, if nature were made of D amino acids instead of L amino acids, we would see sort of the inverted image of this. We would see a beta section here and a large alpha section here and a very small alpha section there. I feel like that's probably too in-depth. We've probably gone too far, so my apologies. What else? So let's talk about the individual amino acids now, because I do want you to know the structures and the names and the one-letter codes for the amino acids. People are going to ask about three-letter codes. I'm like, those are easy. If you know the names, you basically know the three-letter code, but the one-letter code is a little bit trickier. I want you to know them. Why do I want you to know them? Because I care about them, and therefore so should you. The other thing is that when you take biochemistry, your focus is going to be on larger biomacromolecules, but I always want you to remember what the atoms are doing, and the one-letter codes for the amino acids, along with their structures, are how you're going to do that. Yeah?
Up to this point, what's a test question
Up to this point, what is a test question I would ask? In this section, in a lot of ways, let's see, when I've asked questions about peptides and proteins before, I often give you data and ask you to make or draw conclusions. So I don't know. Let me think about that. You should know what phi and psi are. Clearly, you're not going to know how to measure them because that's math, and we don't do math here, right? Can you count? Can you divide by 2? That's generally good enough. So you should know what phi and psi are. So stay tuned. I mean, at this point, the only test questions I can ask are boring, right? Which of the following is the definition for phi? Right, I mean, great, you should be able to answer it. I'm not claiming it's interesting to be able to answer that, but yeah. Yeah. Okay, so let's talk about the individual amino acids. You can group them in various categories. They have different properties. They're all going to have the same backbone structure, and they're going to vary in the identity of the side chain. Now, I'm using squiggly lines because we're talking about an amino acid in the context of a larger peptide or a protein. You need to know the name. You need to know the, one-letter code. And you need to know the side chain pKa. Which ought to be pretty easy because I give you a pKa table, but. All right, so you have some amino acids where the side chain is nonpolar. And there are a couple different categories here, though, and the categories aren't going to be perfect, but we'll sort of do what we can. Alanine is one of the simplest. Its one-letter code is A. And the side chain is just a methyl group, okay? So the thing over here is the alpha carbon. The side chain for alanine is a methyl group. More we could say. Alanine has pretty high helix propensity, which means it's found often in alpha helices. There is more I want to say, but I don't have the data, and I've said it before in previous semesters, and people didn't like it, so no, I just don't have the data to show you. So you can look up Ramachandran plots for each of these individual amino acids, and you can see that they have different preferences. And maybe I'll show you those. I'll have to, maybe during the break I can find those data and show you. But valine is like alanine, only we're going to add more things to the beta carbon. In alanine, the beta carbon is just a methyl group. In valine, you've got two other methyl groups. These are gamma carbons, I guess. Valine is an example of an amino acid that we call, that is beta branched. That is, the beta carbon has two other things on it. That has some consequences for structure. Leucine is like valine, only we're going to insert a carbon between the alpha carbon. We're going to add another carbon in here, so the branch point is going to go back further. So here's the squiggly line. Here's a beta carbon for valine, and I'm sorry, for leucine. The gamma carbon is here, and then you have methyl groups out here at the sort of delta position. And these are all nonpolar. The kinds of non-covalent interactions they can engage in are just sort of van der Waals or dispersion interactions. Isoleucine is an isomer of leucine. Are you surprised by that? Isoleucine has a branch point at the beta carbon, and the beta carbon in isoleucine is also a stereocenter. Its configuration is S. We've drawn it like this. Again, beta carbon, gamma carbon. Like valine, isoleucine is beta branched, and that has some consequences for structure. Last on our list of nonpolar amino acids is one that's called methionine. Methionine is weird. Its one-letter code is M. So far, no surprises, right? The starting letter of the amino acid is the one-letter code. For methionine, you go alpha, beta, gamma, and then at the delta position is a sulfur, and then a methyl group beyond that. This is a thioether functional group. It's like an ether, only the sulfur's here. Methionine's the claim to fame is that it is encoded for by the start codon. And so in a lot of proteins that are expressed, eukaryotic proteins get processed and the first amino acid often gets clipped off. But for proteins expressed in bacteria, they all start with methionine because that's the start codon. The sulfur of methionine, that delta position sulfur, is oxidizable, and so you can form what's called a methionine sulfoxide. This happens in oxygen. It's basically unavoidable, and it can change the character of the amino acid a little bit. Here is the structure of the methionine sulfoxide. Professor J.C. Price's lab sometimes uses the presence of a methionine sulfoxide to assess how accessible a particular methionine side chain is to solvent, whether it's buried inside a protein or exposed. The more exposed the methionine is, the more likely it is to be oxidized. All right, so those are your non-polar amino acids. Let's talk about aromatic amino acids. I group these together because they all involve aromaticity. Most of them are non-polar, though some of them have some amount of polar character, which is why I group them together rather than try to stuff them in the non-polar category. So the easiest is phenylalanine, which is exactly as it sounds. Phenylalanine, here's the alpha carbon. You've got a beta carbon as though it were alanine, and then on that beta carbon you have a phenyl group, okay? We're gonna use the letter P for something else, so we're gonna use F because it sounds like phenylalanine. One interesting thing about the phenylalanine side chain, and I think I mentioned this in chapter 3 of 351, you look at that hydrocarbon and you want to say, hey, that's a non-polar functional group, and it sort of is, but sp3 hybridized carbons are more sp2 carbons are more electronegative than protons. And so in aromatic rings like benzene, you have I will use the color red for electron rich. You have an electron rich face of the ring because sp2 hybridized carbons are more electronegative than our protons. And then you have an electron poor edge of the ring. Electron poor edge and an electron rich face. What that means is you will sometimes see in proteins, we're looking at a benzene ring on the side. Here is the electron-rich face. You will sometimes see the electron-rich face of one benzene ring packing against the electron-poor edge. And I'm making this, that's part of a side chain, so is that. The electron poor edge of one benzene ring can pack against the electron rich face of another benzene ring. This is sort of a T-shaped stacking interaction. And that's found all the time, especially in the interior of proteins. So as we go through, I'll try to tell you some of the types of interactions that these side chains can engage in. Another type of interaction that can happen with aromatic rings, I'll illustrate with some other ones on the list. Tyrosine is the next one. This is our favorite amino acid at B, Y, U. Tyrosine is just like phenylalanine, only it's got a para-hydroxy group. Okay? And the pKa of that proton, as you know from previous exams, is around 10. As with phenylalanine, tyrosine has an electron-rich face and an electron-poor edge. It's also got this polar functional group on the edge of the ring that you can use to do interesting chemistry. Tyrosine sometimes gets phosphorylated, so you can modify this oxygen and make it part of a phosphoester. And then the next aromatic amino acid is called tryptophan. And we're sort of going to use T and R. Tryptophan, that's supposed to be an N. We're going to use T, R, and Y for other purposes. So at this point, we just use W. And so you pronounce tryptophan as though you were 3 and can't say your R's, and you got it right. Tryptophan, that will help you remember. Okay. Its structure is like tyrosine and phenylalanine in the sense that it is aromatic, but it is a polycyclic aromatic group, and the side chain looks like this. So you've got a benzene ring fused to another ring. This type of ring is called an indole, for whatever reason. Can you prove to yourself that it's aromatic? How many pi electrons are in that ring? 2, 4, 6, 8, 10, right? So therefore it satisfies the 4n plus 2 rule. As with phenylalanine and tyrosine, the electron, the face of the indole ring is electron rich, whereas the edge is electron poor. Indole tends to be more electron rich than benzene, though I wouldn't ask you to be able to predict that. One interesting type of interaction so with the indole ring, you'd predict that this nitrogen would be a hydrogen bond donor, and it is. So as with tyrosine, you've got a proton here that can engage in hydrogen bonding. One interesting interaction that tryptophan can be involved in, here's the nitrogen, nope, yes. So we're looking at a tryptophan ring from the side. I'm not doing a very good job of it. I'm not done yet. So this is the edge of the ring that's sort of coming out towards us, and the face of the ring, as I said, is electron rich. There are a number of different proteins in which positively charged groups this is acetylcholine, and there are proteins in which a positively charged group, like this ammonium ion, sits down on the electron-rich face of a benzene ring in what's called a cation-pi interaction. Yeah, so those are important for the aromatic side chains. All right, so we've got more to do there, but we should take a break, and then we'll pick it back up in 5 minutes or so. All right. So, continuing onward past the aromatic amino acids, we're then going to have some that are cationic at physiological pH, or at least can be. The first doesn't fit nicely into this category, but we're going to sort of put it here anyway, is histidine. You could put this in with the aromatic amino acids, though it's more polar than they are, so it sort of fits better here. Here's the structure of histidine. You've got the alpha carbon, a beta carbon, and then branching off of that beta carbon you have this side chain, which I'm going to show you in its positively charged form. In this current form, it is called, that side chain is called an imidazolium side chain. The pKa of the positively charged, of the proton on the positively charged nitrogen is 6, which means that at physiological pH, you've got a mixture of the two forms of the histidine side chain. You have the neutral form, and you have, in which the neutral form we would call imidazole, and then you've got the positively charged form, imidazolium. And pKa's of side chain amino acids are actually highly sensitive to environment. Histidine is often found in the active sites of enzymes, where it acts both as an acid and as a nucleophile. sometimes as both during the same mechanism at different steps. You can see that, is that side chain aromatic, non-aromatic, anti-aromatic? That's an aromatic side chain, and you can tell because it's got 2, 4, 6 pi electrons, and it's fully conjugated. It's aromatic either way. Notice that the side chain, that only one of the 2 nitrogens is basic, right? This was a question in the aromaticity chapter. I can't remember what number was, but the lone pairs on this nitrogen are in an sp2 type orbital, orthogonal to the pi system. They're the ones that are nucleophilic and basic. If you protonated this nitrogen, you would break conjugation, break aromaticity, and that's unfavorable. Okay, so that's histidine. Also, yes?
So this, what you said, it would be a mix of this, the positively charged one and the negatively charged, even though at pH 7 it should be all.
Yeah, so well, pH is a logarithmic scale.
Right.
So comparing a pKa of 6 to a pH of 7-ish would tell you maybe 10 to 1 neutral to positively charged.
Okay.
Though, again, because P side chain pKa is sensitive to environment, depending on what the other amino acids are in the vicinity of this one, that pKa could be much closer to physiological pH, and you might have a 50-50 mixture. Yeah. Okay, we got 2 other side chains that are cationic. One is lysine. Here we've run out again of letters. We already used L and Y for stuff, so K is the letter for lysine. The biggest challenge with lysine is just remembering how many carbons are in the side chain because it's kind of long. So alpha, beta, gamma, delta, epsilon, and then there's an amino group. And the pKa of that ammonium group is 11, which means the lysine is almost always positively charged at physiological pH. There's enough of the neutral form to do some chemistry, but it's fine if every time you see a lysine side chain, you say, okay, that's likely protonated and positively charged. Let's see, and then last on our list of cationic or positively charged amino acid is arginine. Biggest problem with arginine is figuring out how it differs from lysine, because it also has kind of a long side chain with, it's got beta carbon, gamma carbon, delta carbon, and then attached to that delta carbon is a functional group that we haven't said a lot about. It's called a guanidinium functional group. Exam question, final exam question. Which of the following atoms share the positive charge on the nitrogen? All of them, all of the nitrogens do, right, by resonance, okay? So the positive charge in the guanidinium side chain is resonance stabilized. The proton that can be removed to give you neutral arginine is the one that's indicated there. Its pKa is 12, and so with arginine, it's always going to be positively charged. You should think of this functional group as one big group all together. Some people look at that and get confused. They say, oh, I've got 3 amino groups there. No, you have one guanidinium group because the positive charge is shared by resonance on all of those nitrogens. Okay, though, yeah. Sorry.
One more time, which hydrogen would be the one?
Well, you tell me, which hydrogen could I remove and have the molecule be neutral?
On the top one.
Yeah, only the top one.
Or the side, right? By resonance is the same.
Yes, actually, yes. These 2 are identical by resonance. yeah. You could draw all the resonance structures. That's a good point. Sorry, I missed that. You could draw the resonance structures in which, by moving lone pairs to form a new pi bond with this central carbon, you could move the positive charge and have it here, and then these protons would be the acidic ones. You could also, yeah, sort of do the same thing there. All right. Arginine is the pirate's favorite amino acid. Not really even worth a chuckle. So we get to the anionic amino acids. These are going to be negatively charged at physiological pH. At this point, we run out of relevant letters, and so these are going to be difficult to memorize because the letters have nothing to do with the names. Aspartate, I'm giving you its name in its conjugate base form. In its acidic form, it would be aspartic acid, letter D. No real reason behind that, only we run out of others. The side chain for aspartic acid, here's the alpha carbon. You've got a beta carbon, then immediately attached to the beta carbon, you've got a carboxylic acid group. And then, of course, it's going to be negatively charged at physiological pH, but I'm going to have to draw its conjugate acid so that I can write the pKa, which ends up being around 4. Glutamic acid, or glutamate, has the one-letter code E. I guess at least the letter E is in the name of glutamate. Glutamate differs from aspartate in the addition of a carbon in the side chain. So for glutamate, the carboxylic acid is on the delta carbon rather than the gamma carbon. But as with aspartic acid, glutamic acid similarly has a pKa of about 4. Meaning that at physiological pH, it's almost always negatively charged. Aspartic acid, though, is found in enzyme active sites. pKa apparently changes inside that enzyme active site, and you can access some of the acidic form. Maybe we'll talk about some enzyme mechanisms a little bit later, and I'll show you an example of what aspartate does. Then you've got, yeah, sure.
Do you care that we know like that those pKa's are slightly different? Like, do you care about decimal places or
No. Do I care about, no. pKa is one significant figure. Well, no decimals. I suppose I do want you to know that 10 is different from 11, but yeah, I don't need you to know that 4 is different from 4.5. That's, yeah. Let's see. Then you've got a list of polar amino acids that are neutral. And this is sort of a hodgepodge. One of them is called serine. One-letter code is S, so at least that makes sense. The serine is like alanine, only it's got an OH group on it. pKa of just your regular run-of-the-mill non-resonance stabilized OH group is 16. A relative of serine is threonine. There's where we're using our letter T. Threonine is just like serine, only the beta carbon has a methyl group on it too. This is our third beta branched amino acid. That can influence conformation and shape because it means the beta carbon is more sterically crowded for threonine than it is for serine. Then you also have the cousins, the neutral cousins of aspartate and glutamate, where, excuse me, we're going to keep the same basic side chain structure, only we're going to have amides instead of acids or carboxylates. And so the amino acid asparagine, if you go back and compare its structure to aspartate, it is exactly the same, only you've got an NH2 instead of an OH. The one letter code for asparagine is N. Again, doesn't necessarily help you out too much other than I guess there is an N there. And then the analog of glutamic acid is just like asparagine, only there's an extra carbon in the side chain. So now the carbonyl group is on the delta carbon rather than the gamma carbon. And here we throw out all pretenses and we just choose one of the remaining letters, and it's Q. So there you go. Lastly is on the polar but neutral amino acids is a unique one, and this is called cysteine. Before I draw cysteine, I guess I will say that serine, threonine, and asparagine all can be post-translationally modified. PTM stands for post-translational modification by something called glycosylation. That means we can covalently attach a sugar to either the OH group or the NH group, and that can dramatically change the structure of the side chain and its function. I haven't pointed out all the other places of the other amino acids that can be post-translationally modified. That's a subject for another time. So we are going to talk about carbohydrates, though, in the near future, so I just thought I would point that out. That's not anything to memorize. Cysteine, one letter code C, at least it makes sense. Cysteine is the sulfur version of serine, so you're going to have an SH instead of an OH. And cysteine is a really good nucleophile, so you'll see it as a good nucleophile in enzyme mechanisms, but it's also oxidizable. And one of the most common forms that you'll see for oxidized cysteine is what's called a disulfide. This is where two cysteine sulfurs are bonded to each other. And if you think about those two, if you think about starting with two cysteine side chains and you look at what's missing between the two cysteine side chains and the disulfide, you're missing 2 protons and 2 electrons. That's an oxidation reaction, okay? Cysteine disulfides form spontaneously in air, and yeah, so the disulfide is the oxidized form, and just the free thiol is the reduced form. What else to say there? Disulfides are important for structural integrity. It's a cross-link between two different positions on a protein that can provide some structural rigidity. In case you want to know, I don't know that this would be important to you in the immediate future, the interior of the cell, the cytosol, is a reducing environment. The exterior of the cell is an oxidizing environment. So there aren't any disulfides generally inside the cell, though there are disulfides on extracellular proteins and membrane proteins. Antibodies, as an example, are highly disulfide bound, and a lot of proteins that come through the secretory pathway, that is through the ER and the Golgi and then get transported outside the cell, a lot of them are highly disulfide bound. Lastly is the weird ones. You didn't think that everything else was weird, but the last two are just sort of strange, so they don't really fit anywhere. One is glycine, and I'll just draw the entire amino acid because glycine doesn't have a side chain. You're like, buddy, couldn't you have led with that one? Okay, so no side chain. Because of that, glycine is flexible. And it doesn't have the same steric problems. If you go back to that Ramachandran plot that we were doing, glycine doesn't have the same steric problems as the other amino acids do. And so it's more comfortable in a diverse and broader range of environments. The next is proline, and proline has the one-letter code P. Proline is weird because the side chain actually bites back on the amide nitrogen such that the amino group in the proline amino acid is a secondary amine rather than a primary amine. And in a peptide or a protein, proline doesn't have an NH proton. Okay? One of the interesting things about proline is, well, there's two. First, if you think about that phi angle, there are some values for phi that are not allowed for proline because it's in a 5-membered ring. In other words, you can't rotate completely freely around that bond because you have the ends are connected to each other. Go back, if we think about the phi dihedral angle that we were talking about, for proline, this proton and this R group are attached to each other. So that constrains the values for the phi dihedral angle that proline can adopt. The other interesting thing about proline is if you go back here to the omega dihedral angle, do you remember we said that the trans amide is generally more stable than the cis amide because sterics, right? But for proline, this proton there is just something else, right? It's just a carbon. So for proline, there's not a big difference in stability between cis and trans amide configurations. And so you can find proline in both of those situations. What do I mean by that? Let's just illustrate with, I'm going to draw proline, oops, except I can't draw a 5-membered ring.
It's always 5, right?
Yeah, always 5. So this is that omega angle, right? This is the trans configuration. This is the cis configuration. Can you see in terms of sterics, there's not a whole lot of difference between those two situations, right? With a regular amino acid, this is a hydrogen, and so the trans configuration has way less steric hindrance than the cis configuration. But with proline, both of the things that are attached to the amide nitrogen are carbons. So, as my dad used to say, it's about a horse apiece. I don't know what that means other than it's the same thing either way. And so you can find proline in both of those environments. Because it lacks the NH group that all of the other amino acids have, proline is different and it doesn't fit into a lot of the same secondary structural environments. We haven't talked about the alpha helix and the hydrogen bonding pattern of an alpha helix. We're going to do that now. But proline doesn't fit very well within an alpha helix because it lacks the key hydrogen bond donor. All right. That's your whirlwind tour through the 20 amino acids. Good luck memorizing stuff.
Is that pretty much all there are? Or there are a lot more?
Are there others? Yes. Yes. I will say more about that, but go ahead.
On the study guide, it says the PTA for S and T. Is it are they both 16?
Yeah.
Okay.
Yep. Sorry about that. They're both 16. Yeah, there are lots of other amino acids. For example, nature actually at some rate incorporates selenium, which is one row down from sulfur, in the presence. So it incorporates selenium instead of sulfur. So there's selenomethionine and selenocysteine. I don't know a lot about that. There are tons of other unnatural amino acids that are synthetically useful that we haven't talked about here, but could. But these are the ones that nature codes for. So this is the alphabet. Yep. Yes? Yeah.
And could you say in what details you want to have memorized for this being able to recognize
Structure,
what his name is?
structure, name, one-letter code, pKa. Okay. So now we need to talk about something called protein folding. And that is, what is the relationship between sequence of amino acids and then structure? And this is an important problem. It's one that's very difficult to solve. It's one that actually, within the past couple of years, computer scientists have brought to bear all the power of artificial intelligence to understanding sequence and structure. And there's some really neat tools now available, but it's still an interesting problem. So I want to illustrate this by giving you an amino acid sequence, and then we're going to talk about the structure that it forms, okay? So hang in there. I'm going to give you a one letter code, and this is 30 residues long, 30 amino acids long. Oops. And when you write out a sequence, sometimes it's good. I'm writing this out in the direction N to C, which means this side of the protein has a carboxylate group, and this side would have an ammonium group. All right. So, chemists can now look at that sequence and they can pretty well tell you what shape it should form. And there are some rules for understanding that. And we're going to learn some of what those rules are. It's not as bad as you think. What? All right. Let's start out by principles of protein folding. You've got basically a couple of things going on here. You've got the hydrophobic effect. You learned about that in 351. This means there is a penalty for having non-polar functional groups exposed to water. Remember learning about that? Oil and water don't mix. Hydrophobic effect means there's a penalty for having non-polar groups exposed to water. We don't have time to talk about that in a lot of detail now. You may want to go back and review chapter 3 and 351 if that sounds unfamiliar, because we're going to rely on it to rationalize why stuff is going to happen. And then a second major theme is non-covalent interactions. That is things like hydrogen bonding, ion-ion interactions, which we're going to call here salt bridges. That's a term from biochemistry, but that refers to a positive charge interacting with a negative charge. Okay. So let's go ahead and identify non-polar side chains here. Go back and look at your list of amino acids, and let's try to highlight, I guess, in I'll highlight in orange all of the nonpolar amino acids. All right, so what do you see? L is leucine. So the leucine side chain would look like that. The V side chain would look like that. V is easy to remember because it looks like it. Yeah, all right. So we got L's. Okay. N is N. M. It's always hard to hear the difference between M and N. Okay, good. A, it's hard to know. A is sort of a methyl group, so it's not really super nonpolar. It's sort of just meh. Okay, so we've got nonpolar amino acids. Let's highlight positively charged ones. Let's use blue for positively charged. What do you see? Okay. R and K, good. H will depend on pH, right? So not totally clear what we should do there. It depends on, we could probably guess that at physiological pH it's mostly not going to be protonated. Did I miss any of the Ks? I missed an R, right? All right, and then let's use red for negatively charged. Got E's, more E's. Final exam question. What is the net charge on this peptide? At pH 7. 1, 2, 3, 4, 5, 6, 7, 8, 9 plus. 1, 2, 3, 4, 5, 6, 7, 8 minus. Okay. Now, as I said, there's a driving force in water to have nonpolar functional groups hidden from water because of what's called the hydrophobic effect. It's entropically driven. It involves releasing ordered water molecules to be free in bulk solvent. Let's see. So we want to have an idea of how the backbone of this protein, coming back here, what phi and psi angles could be adopted that would cause the nonpolar side chains to be away from the solvent. So as you look at these nonpolar side chains, there is a characteristic spacing between them. It's not perfect, but it applies a lot. For example, nonpolar methionine is 3 residues away from nonpolar leucine, which is then 1, 2, 3, 4 residues away from valine. So there's sort of this spacing of 3 amino acids and then 4 amino acids. And then it sort of follows again. There's a 3 amino acid spacing, let's see, here there's another 3 amino acid spacing and a 4 amino acid spacing. And there's, as I said, it's not perfect. There's a 3 amino acid spacing there. There's a 4 amino acid spacing there. There is a secondary structure you can adopt that would cause all of these nonpolar side chains to be on one side of the molecule. And to show you this, what this secondary structure is, I need to introduce to you the alpha helix. So let's see, I need to download, sorry. Probably we're going to need more than 1 day on this because I just keep talking. So we have some flexibility in the schedule. I'll generate an updated syllabus schedule based on what I think is going to happen. Next time we are probably still going to be on peptides and proteins because we want to finish this discussion, and then I need to tell you how we make peptides and proteins in the first place. All right, sorry, is that it? Yes. Behold, the alpha helix. All right, so this is the backbone of an alpha helix. I'm not showing you any side chains. Nitrogens are shown in blue, carbons are shown in yellow, and oxygens are shown in red. Okay? One interesting feature of the alpha helix, if we start sort of counting here and call this the I position amino acid, there's a nitrogen, an alpha carbon, a carbonyl carbon. Then this one would be I plus 2. Sorry, 1. Counting doesn't work anymore. Yeah. This would be I plus 2. This would be I plus 3. And then this one up here would be I plus 4. Notice that the amide nitrogen of the I plus 4 amino acid is hydrogen bound to the carbonyl oxygen of the I position amino acid. So that's called I to I plus 4 hydrogen bonding between a carbonyl oxygen and an amide proton. What does that do? And that's a regular pattern. You can see it all along the way. So if we call this the I position amino acid, then 1, 2, 3, 4 amino acids away, we have a hydrogen bond right there. Now, does that make sense? Can you see that? Okay. That regular pattern of hydrogen bonding of an I position carbonyl oxygen to an I plus 4 position amide nitrogen makes it so here is where the R group would be for the I position side chain. Here is where the R group would be for I plus 3. Here is where it would be for I plus 4. This pattern of hydrogen bonding makes it so that the I position side chain and the I plus 3 or I plus 4 side chains are all close to each other, right? They're on the same side of the helix. All right. And if we actually look down an alpha helix, no, do we want to do that yet? Sure. Trying to decide. If you sort of summarize alpha helical structure, you can say there's basically 3.5 to 3.6 amino acids per turn of the helix. And that's reflected in the fact that the I position carbonyl oxygen is hydrogen bound to the I plus 4 position amide nitrogen. By the time you get around here, you're between 0.9 and 1 and 1.1 turns of the helix, okay? So each turn of the helix houses 3.5, maybe 3.6 amino acids. That means every 3.5 amino acids, you're basically wrapped around the helix back to where you started, only further down the helix, right? So let's imagine what would happen if we look at that sequence that we wrote. If we form an alpha helix, by the time we get to 3 or 4 amino acids away, we're basically on the same side of the helix, right? So that would mean twist back to the same spot, twist back to the same spot. Suddenly, if you fold into a helix, you've got a stripe of non-polar amino acids all occupying one face of the helix. Now we're going to take a different view of this. Here is the sequence that we just looked at. And we're going to look down we're going to put our eyeball here, and we're going to look down the axis of the helix from the N terminus of the peptide looking towards the C terminus. And what we would see is something like this. And I'm showing you 7 amino acids, and I'm giving each of these amino acids one letter of a 7-letter sequence starting with A. Position A, B, C, D, E, F, and G. And you see that as I go from A to 3 amino acids away, B, C, and D, I'm back to sort of the same face of the helix, okay? And if I look at that sequence that I have, this is sort of zooming in on the peptide that forms from this structure. A position is a methionine, which is non-polar. And then D position is a leucine, which is also non-polar. If I map this sequence out onto an alpha helix, I have the 2 non-polar groups on the same side of the helix with polar groups at the other positions. Okay? Questions you want to ask about that? I feel like this makes sense to me because I've been staring at something like this for about 20 years, but this is maybe the first time you've seen it, so it's fine if it doesn't make sense, and I'm not sure I explained it very well. So, questions?
So all those side chains that are sticking out of the helix, these are our non-polar side chains?
These 2 are non-polar. This is a lysine. So the funny thing about this is a, I should point out that blue atoms are nitrogen, that red atoms are oxygen, that yellow atoms here are carbon, and I should also point out that this is derived from what's called an X-ray crystallography experiment, and X-ray crystallography of large proteins doesn't see protons. So I've left off all of the protons. So this is a lysine side chain, which would be positively charged. This one is glutamate. This one is, glutamine, Q with a nitrogen. This is glutamate, which would be negatively charged. This is another lysine side chain, which would be positively charged. This is a glutamate, which would be negatively charged. This is a leucine, this is a methionine. We're mapping these first 6 or 7 amino acids onto the alpha helix. Okay, other questions? What you've got here, if you fold this sequence into an alpha helix, is you have something that's what I call globally amphipathic. That means it has a nonpolar face and a polar face. Now you've seen molecules like this before. We talked about surfactants that had a polar head group and a nonpolar tail. We talked about how those surfactants self-associate to bury the nonpolar tail inside of a micelle with the polar head groups exposed to water. We're going to do something similar with this sequence. It's going to form what's called a coiled coil, where the nonpolar side of one helix packs against the nonpolar side of another helix to keep all the nonpolar stuff away from solvent and the polar stuff exposed to solvent. And this is what's called a coiled coil or a helix bundle, and you will later learn in biochemistry of similar types of arrangements that are called leucine zippers. We'll have more to say about that a little bit next time, and then we'll talk about peptide synthesis.